Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled, Challenges and Considerations in Clinical Development of Targeted Therapies, AML as a Case Study. My name is Candace, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Medpace, who develops the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing Phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas. Now I'd like to introduce our speakers for today's event. Dr. Gregory Hale is a physician with over 26 years of experience in all phases of clinical development, with expertise in hematopoietic stem cell transplantation, cellular and gene therapies, and immuno-oncology. Dr. Hale has served as clinical director of the Transplant and Gene Therapy Program at St. Jude's Children's Research Hospital and medical director of the Division of Hematology Oncology at Johns Hopkins All Children's Hospital. He has authored more than 200 peer-reviewed manuscripts, review articles, and book chapters. Our next speaker is Dr. El Mustafa Bahasi, who received his PhD in molecular biology and biotechnology from the University of Brussels in Belgium, after which he joined the University of Texas Southwestern Medical Center in Dallas as a postdoctoral fellow in molecular oncology. Following his training at UT Southwestern, Dr. Bahasi moved to the Department of Cancer Biology at the University of Cincinnati as a research scientist and later on as a faculty member in the Division of Hematology Oncology. As an independent faculty, Dr. Bahasi developed a translational line of research where he worked closely with clinical oncologists to develop new companion diagnostics using cutting-edge genomic technologies. And our final speaker for today is Dr. Jeffrey Vassallo, who has a PhD in molecular biology with a postdoctoral training in experimental hematology. He has worked in drug development, including preclinical and clinical research at large pharmaceutical companies for more than 24 years. During his tenure at Medpace, Dr. Vassallo has managed global Phase 1 through 3 trials in hematology and oncology with an emphasis in advanced therapies. This diverse experience gives him a unique perspective on the operational aspects in the clinical development of novel therapeutics for AML. And now, without further ado, I'd like to hand the mic and the presentation over to our speakers, who may begin when they're ready.
Welcome. Targeted therapies have been increasingly explored in the fields of oncology, largely because of the anticipation that they will provide improved efficacy or disease control while also reducing toxicity. A lot of the initial work has been done in the solid tumor arena, but today we'll focus the discussion on targeted therapies for acute myeloid leukemia. Acute myeloid leukemia is an aggressive malignancy that arises from the clonal expansion of immature hematopoietic cells that are arising in the granulocytic or the monocytic lineage. Typically, these patients are quite ill at diagnosis with pancytopenia that can contribute to infection due to neutropenia, hemorrhage due to thrombocytopenia or low platelet counts, and anemia due to low hemoglobin. Acute myeloid leukemia may arise de novo as the initial malignancy a patient experiences. It may occur due to prior exposure to radiation, environmental toxins, or chemotherapy and be classified as therapy-related, or can arise following transformation from another malignant or pre-malignant disorder, such as myelodysplasia or myeloproliferative neoplasm. Acute myeloid leukemia, or AML, is the most common leukemia in adults with a median age at onset of 70 years. In 2019, it's anticipated that there'll be 21,000 new cases of AML diagnosed in the United States. The World Health Organization in 2016 provided a revision of the classification of acute myeloid leukemia. An example of it is shown here. As you can see, most cases of acute myeloid leukemia are defined by recurrent genetic abnormalities, but there's also a variety that are associated with a variety of appearances under the microscope. In 2017, the journal Blood published the work of the European Leukemia Net group, which provided recommendations for the diagnosis and management of patients with acute myeloid leukemia, and has also provided mechanisms for disease assessment. One thing that's evident from these classifications is that over time, acute myeloid leukemia has become better defined from rather one single entity, but has been recognized as consisting of a variety of subgroups based on cytogenetic and morphologic criteria. One important point here is that acute promyelocytic leukemia is a unique circumstance which has a different prognosis, largely due to completely different treatment based on its cytogenetic abnormalities. The translocation between chromosome 15 and 17 will juxtapose the PML and RARA genes in close proximity, and therefore agents such as ATRA, all-trans-retinoic acid, and arsenic trioxide are effective in treating this disorder, largely avoiding the need for chemotherapy. It's been one of the early successes for targeted therapies in oncology. There are many changes in the genome or the hematopoietic cell that contribute to myelodysplasia or acute myeloid leukemia. The MDS-AML continuum is a spectrum with AML representing the full malignant expression of the abnormality. It's not uncommon that AML can arise on a background of myelodysplastic syndrome, but many of the changes that occur result in abnormalities in apoptosis, which is programmed cell death, or changes that result in a leukemic, in a proliferation or growth advantage of the leukemic clone. In addition, there can be alterations in histone acetylation, as shown on the right-hand side of the slide, leading to epigenetic alterations, which result in differences in genes being transcribed or available for transcription. There can be alterations in the immune environment, leading to the ability of cytotoxic T cells or natural killer cell mediation that are ineffective in eradicating or recognizing leukemia. Certain changes in the bone marrow environment, chromosomal instability, as well as environmental factors can result in this malignant degeneration. Acute myeloid leukemia is rapidly fatal without therapy, with a life expectancy on the order of weeks. Acute myeloid leukemia typically requires intensive chemotherapy, initially called induction therapy, with subsequent maintenance chemotherapy, with consolidation with hematopoietic cell transplantation for high-risk patients. Death from disease or from treatment-related complications occurs too commonly, and the 5-year overall survival rates typically vary by patient age. For young adults, which are typically those under the age of 60 years, the overall survival rates approach 40%, and in older adults greater than 60 years of age, the overall survival rate is significantly lower, approaching 10%. Certainly, there is a high unmet medical need for therapies that are able to control acute myeloid leukemia for this patient population. In the 1970s, the standard regimen consisting of cytarabine along with an anthracycline for induction was introduced. That was typically referred to as the 7+3 regimen. There have been no significant improvements until the last 1 to 2 years in the type of therapy recommended for these patients. In choosing patients for an AML therapy trial, considerations include excluding or separating AML in 3 patients, which are acute promyelocytic patients. As we discussed earlier, these patients have a different cytogenetic abnormality, which allows them to have a higher cure rate and a better outcome with significantly less intense chemotherapy or less intense therapy, which is largely targeted to their cytogenetic translocation. AML is typically defined by risk groups, as we have mentioned earlier with the European Leukemia Net group providing a good overview in the 2017 Blood article. And certainly patient age and patient condition is a consideration, with older patients having worse outcomes than younger patients. Induction chemotherapy most commonly consists of cytarabine with an anthracycline, such as daunorubicin, although in older patients, we've recently seen in clinical trials that targeted therapies with venetoclax with a hypomethylating agent may result in improved outcomes, and we'll discuss this later. As we'll see later, there's also new agents that combine cytarabine and anthracycline in a novel combination, such as the liposomal formulation, as we'll discuss later with the development of the compound CTX-351. And finally, post-remission therapy is important and consists of either intermediate doses of cytarabine for low-risk patients or those who are medically unfit for transplant, or allogeneic hematopoietic stem cell transplant from related or unrelated donors for those patients with high-risk disease. Finally, supportive care is critically important in this group of ill patients. These patients are typically ill from the time of diagnosis and become more ill during their course of intensive chemotherapy, so that infection, bleeding, and organ dysfunction remain significant causes of morbidity and mortality. Typically, these patients require aggressive transfusion support as well as infection prophylaxis and surveillance. Over time, the molecular and genetic basis of acute myeloid leukemia has been better defined so that we're able to recognize different risk groups, so that it's actually a heterogeneous group of diseases rather than a single entity. Risk stratification is critically important, and as I mentioned earlier, that involves not only the patient's age, but also the genetic and molecular basis of the patient's malignancy. And finally, risk-adapted therapy becomes important, and we'll talk about some of the ways that we can develop targeted therapies by either targeting a specific genetic abnormality, a specific biochemical pathway, or a specific AML subset. It is worth noting that in the last 3 years, the Food and Drug Administration has approved 8 new targeted agents for use in the treatment of acute myeloid leukemia in the United States. It is equally important to recognize that none of these, however, have yielded as promising results as we've seen for ATRA or arsenic trioxide in acute promyelocytic leukemia, or for the tyrosine kinase inhibitors such as imatinib for chronic myeloid leukemia. Targeted therapies are defined as cancer treatments that target the cancer-specific genes, proteins, or tissue environments. Much of the early work has been done with small molecule drugs and monoclonal antibodies in the solid tumor arena, such as in breast cancer, colon cancer, lung cancer, and melanoma. In order to develop a targeted therapy, a molecular knowledge of the cancer must be undertaken, and you must be able to match the targeted therapy to the patient's specific abnormality. Some of the therapies may be tumor agnostic or site agnostic, such as looking for specific expression of proteins such as the NTRK fusion protein, which is evident in lung, colorectal, and pancreatic cancer, and expressed to a higher extent in other cancers such as thyroid cancer and infantile fibrosarcoma. When developing targeted therapies, it's important that patients be observed for off-tumor toxicities, which would be on-target off-tumor toxicities, as these can be significant and are typically different from traditional cancer therapies. It largely depends upon the specificity for the target as well as the distribution of the target on normal tissues. Of course, these agents can be used alone or more commonly in combinations with chemotherapy and radiation therapy to enhance the disease control. Finally, oftentimes, with the development of new targeted therapies, the development of a companion diagnostic assay may be necessary as well. When identifying targets in any malignancy, but especially acute myeloid leukemia, we do have a variety of mechanisms to do this. This includes the use of cytogenetics, flow cytometry, molecular analysis, next-generation sequencing, in a variety of measures. Many of these are provided as standard of care, while others may not be typically provided in a standard of care environment and can be considered investigational as part of the trial. It is important to note that cellular abnormalities often occur in combination, so you may find several cytogenetic or target abnormalities present in a patient's tumor, and it's not uncommon to find abnormalities that are of undetermined clinical significance so that you're not able to comment whether or not they are contributing to the patient's AML. Of the agents that have been approved by the Food and Drug Administration for treatment of AML in the last 3 years in the United States, they are highlighted here. The ones in red are the agents that are what we would consider targeted therapies based on our current description and definition. CPX-351 is not highlighted. It is a combination of chemotherapy drugs that are modified to be provided in a 5:1 molar ratio in a liposome. So it's provided in a very specific way to enhance its uptake to the malignant cell, but is not a targeted therapy by the definition that we typically would use. Next slide. As you can see, 4 agents were approved in 2017. 4 agents were approved in 2018. The points that I would emphasize on this slide would be the first agent, midostaurin, is a FLT3 inhibitor, and it was the first tyrosine kinase inhibitor approved for acute myeloid leukemia, and the first approved for a mutation-specific form of acute myeloid leukemia that was not acute promyelocytic leukemia. So it was one of the first forays into targeted therapies for acute myeloid leukemia. Gemtuzumab ozogamicin we'll talk about later. It is a CD33 targeting agent that also has a payload of calicheamicin, which is a toxin that will be delivered directly to the cell. It has the unique history of being approved by the FDA, then withdrawn, and then reapproved at a later date. Other agents that are important to note here include venetoclax and glasdegib. These agents are approved for the treatment of AML in combination with other chemotherapy agents. Next slide. As you can see here, targeted therapies are typically categorized for the purposes of this discussion into 3 groups. The first is mutation-targeted agents. So once we know a specific agent that is oncogenic in the development of acute myeloid leukemia, such as FLT3 or isocitrate dehydrogenase IDH inhibitors, those would be considered in this group. Second, there are non-mutation targeted novel agents, and these are agents that work through an epigenetic mechanisms or that control or alter apoptosis. And finally, those agents that involve the targeted delivery of cytotoxic agents, such as antibody-drug conjugates. The first group we'll talk about will be those that target specific mutations. FLT3 inhibitors were the first class of AML inhibitors or targeted therapies that have been used outside of acute promyelocytic leukemia. FLT3 stands for FMS-related tyrosine kinase 3, and it is a tyrosine kinase that is found on hematopoietic cells. It's actually one of the most common genetic abnormalities that are identified and is expressed to a lesser extent on hematopoietic precursors such as CD34 positive stem cells, and is expressed less commonly with maturation. Approximately 30% of patients with acute myeloid leukemia will have a FLT3 internal tandem duplication mutation, with 5 to 10% of these patients having a tyrosine kinase domain mutation noted. FLT3 internal tandem duplication, or FLT3-ITD, is associated with a worse prognosis as well as a higher white blood cell count at diagnosis and higher allelic ratios. Comparing the abnormal FLT3 with the wild type FLT3 are also associated with a poor prognosis and higher relapse rate. Prognostic significance of this gene abnormality can also be altered when looking at the patient's age, but also by other mutations that are present within the patient's leukemia cell. For example, coexisting NPM1 mutations in patients who have FLT3-ITD may have a better prognosis than those who lack that mutation. The first agent that was developed as a FLT3 inhibitor commercially was midostaurin. It is a tyrosine kinase inhibitor that's also active against a variety of other kinases such as VEGF, platelet-derived growth factor, and KIT. Its approval was based on the RATIFY trial, which was a randomized clinical trial in which patients were randomized between an induction regimen of 7 plus 3 plus placebo or 7 plus 3 plus midostaurin, and then maintenance therapy consisted of midostaurin or placebo administered for approximately 1 year after remission was obtained. The RATIFY trial only enrolled patients under the age of 60 years and demonstrated a 23% reduction in the risk of death. Midostaurin's effect was enhanced with chemotherapy as opposed to when it was given as a single agent, and is typically given on days 8 through 21 of each cycle at the end of consolidation. In addition, midostaurin is also now frequently given as part of post-remission therapy, including after transplant, although this is not yet as clearly proven to have a clinical benefit as it has been when given after induction. The second FLT3 inhibitor that has been recently approved in 2018 for use in the United States for treatment of AML is gilteritinib. A randomized clinical trial of this agent showed an improved complete remission rate of 34% versus 15%, with a 36% reduction in death at 1 year. Gilteritinib has been studied in combination with a variety of agents, such as hypomethylating agents, such as azacitidine and azacitidine, low-dose Ara-C, other chemotherapy agents such as mitoxantrone, as well as fludarabine and Ara-C. Gilteritinib is a much more potent and more specific FLT3 inhibitor than midostaurin. It's uncertain how it will fit into the use of a FLT3 inhibitor, especially for patients who previously received midostaurin. Newer generation FLT3 inhibitors are also being evaluated given the common occurrence of FLT3 abnormalities in AML patients. They include crenalinib and quizartinib. Certainly these agents are being studied alone and in combination with other chemotherapy agents. In this class, the most common adverse events are those that are typical for AML therapy and include nausea, vomiting, fever, febrile neutropenia, dermatitis, headache, and bleeding. One note is that the identification of FLT3 naive patients, that is patients who have not received FLT3 inhibitors, will be increasingly difficult as newer agents are developed in this class given the use of midostaurin and gilteritinib. The next group of mutations that we'll talk about will be isocitrate dehydrogenase, or IDH inhibitors. IDH currently exists in 3 forms and is responsible for the oxidative decarboxylation of isocitrate to alpha-ketoglutarate. This results in the accumulation of 2-hydroxyglutarate within the leukemic cell that leads to DNA hypermethylation via inhibition of histone demethylation. This is an epigenetic mechanism of action, and it results in a block in cell differentiation. These mutations are much less common than FLT3 mutations. IDH1 mutations occur in about 10% of AML patients, and IDH2 mutations occur in about 9 to 13% of acute myeloid leukemia patients. While FLT3 mutations are clearly associated with an inferior outcome, IDH mutations are considered prognostically neutral. They do not indicate an improved outcome or a worsened outcome, but they do represent a target for therapy. Ivosidenib is an agent that targets IDH1 and has received commercial approval. Its approval was based on a phase 1 study which demonstrated an objective response rate of nearly 42% with a composite complete remission rate of 30.4%. A composite complete remission rate for acute myeloid leukemia typically includes patients that have achieved a complete remission or those who have achieved a complete remission with incomplete recovery of platelets. The target for IDH2, enasidenib, has also been recently approved based on the results of a phase 1/2 study and an objective response rate of 40%. Both agents are well tolerated, with a unique adverse event that can occur in about 10% of patients being differentiation syndrome, where the cells actually mature into mature granulocytes, very similar to what we see with atrophy in acute promyelocytic leukemia. Turning to the next group of agents, which are non-mutation targeted agents, such as the BCL2 antagonist. BCL2 is an anti-apoptotic protein, So therefore, it inhibits the ability of cells to undergo programmed cell death. BCL2 has been identified to play an oncogenic role in lymphoid and myeloid malignancies, and therefore venetoclax is the agent that has most recently been approved to target BCL2 and is quite a potent BCL2 inhibitor. The advantage of this group of agents is there's at least a theoretic possibility that they may eradicate leukemia-specific stem cells. However, venetoclax is rarely used alone to treat acute myelocytic leukemia and is typically used in combination, especially in elderly AML patients. Venetoclax is typically combined with azacitidine or decitidine, both of which are hypomethylating agents, and in recent studies have demonstrated a composite complete response rate approaching 70% with a median overall survival approaching 18 months in the elderly population, which is an incredible finding. I would emphasize that when used alone as a single agent in the same population, the complete response rate is only 6% with progression-free survival of 2.5 months, so there's definitely a significant rationale for using it in combination with hypomethylating agents. It can also be used in combination with low-dose cytarabine, but with less promising results. Adverse events are tolerable and are typically what one would expect in this patient population. Moving on to epigenetic modifiers, this includes the agents such as azacitidine and decitidine. These are agents that result in post-translational modification of DNA and histone proteins, and they are typically most commonly DNA methylation, although a variety of other mechanisms are noted and are listed here. Hypomethylating agents typically target DNA methyltransferase and are quite active in treating patients with AML and MDS. Overall response rates in this group approach 50% and can be upwards of 80% for patients receiving combination chemotherapy. These agents can be used alone or in combination with chemotherapy and are increasingly used as maintenance therapy after hematopoietic cell transplantation. Cytopenias and hepatotoxicity are the most common toxicities observed in this group. Non-mutation targeted agents include a variety of agents listed here. The one that I'll talk about the most will be hedgehog signaling pathway inhibitors. Glastigib is the agent that has been approved for treatment of acute myeloid leukemia, and it is an agent that actually inhibits the protein smoothened, which is also active in another malignancy, basal cell carcinoma. Phase 3 trials of Glastigib in combination with cytarabine or with the agent alone showed a 50% reduction of death and an improved remission rate of 17%. This agent has not been compared directly with hypomethylating agents or used in combination with them. It's currently approved for use in combination with low-dose cytarabine in the elderly population or the medically unfit patient population. Its adverse event profile includes febrile neutropenia, musculoskeletal pain, rash, and decreased appetite. As you can see here, there's a variety of other enzymes that can serve as targets for this group. Antibodies such as bispecific T-cell engaging antibodies that are used in acute lymphoblastic leukemia and cellular therapies such as CAR T-cells that are also used in acute lymphoblastic leukemia are being investigated in clinical trials for acute myeloid leukemia as well. And finally, targeted delivery is important. Antibody drug conjugates provide a mechanism whereby the target will deliver a toxic payload directly to a malignant cell. The drug that's recently been reapproved for this group is gemtuzumab ozogamicin. It is a humanized anti-CD33 antibody, which is conjugated to a toxic molecule called calicheamicin. The CD33 is expressed on a variety of granulocytes, being expressed more commonly as the granulocytes mature and less commonly in the hematopoietic stem cell population. Upon binding to the cell, the antibody drug conjugate is internalized and the cytotoxin is released to result in cell injury and death. It was initially approved in the year 2000 for elderly AML patients and subsequently withdrawn in 2010 after subsequent trials demonstrated no significant survival benefit and concerns over higher hepatotoxicity rates in the patients who received gemtuzumab ozogamicin. Subsequently, a pooled analysis of 5 randomized trials did demonstrate improvements as far as a lower relapse rate and higher overall survival rates in favorable and intermediate risk AML patients, leading to FDA reapproval in 2017. As you can see, targeted therapies do represent a challenge. There have been increased interest in developing less intense therapies, presumably less intense therapies, given the targets are specifically for acute myeloid leukemia. One of the questions to address is how these agents are developed alone or in combination with other chemotherapy. It is important to note that while targeted therapies do offer promising results in this patient population, none of these agents has yet achieved the clinical results that we have observed in acute myeloid leukemia in 3 or in chronic myeloid leukemia with tyrosine kinase inhibitors. Certainly, reproducible criteria needed to be developed to avoid selection bias. Given the older age of the onset of diagnosis for this group, geriatric assessment for fitness is often important, and the assessment for comorbidities for participation in clinical trials. Certainly, the field is moving target, as we can see that 7+3 chemotherapy regimen has been the induction regimen for 40 years, but certainly venetoclax with hypomethylating agents has demonstrated a markedly improved outcome and profile, and as the field changes, we'll need to be cognizant of the fact of how we're comparing new agents. And finally, that regardless of the poor outcomes that we see with acute myeloid leukemia, disease recurrence continues to remain the primary cause of treatment failure for this patient population. And now at this point, I'll turn over the talk to my colleague, Dr. Bahasi, who will discuss many of the laboratory parameters and NGS in clinical practice.
All right. Thank you very much. I will start first by introducing NGS, also known as next-gen sequencing or massively parallel sequencing. So many of the important biomarkers for the diagnosis, risk stratification, treatment initiation, and or modulation, as well as for response assessment in EML, were discovered from NGS studies, including whole genome, whole exome, and panel sequencing studies. In NGS, genomic DNA is fragmented into small fragments and then tagged with small adapters, and these tagged fragments are then immobilized on a solid surface called flow cell, amplified, and per end sequenced. During sequencing, a signal is detected when a base is incorporated, and this signal can be in the form of light when a pyrophosphate is released from addition of a complementary base. And this principle is used by the Illumina platforms. Ion torrent platforms use the concept of sequencing by synthesis, where a hydrogen ion is released from addition of complementary base, leading rather to a pH change. So this leads to a generation of short and contiguous reads, and these reads are aligned to reference sequence and analyzed. The analysis is computationally very intense. And once the analysis is done, some variants will be discovered and curated to determine their pathogenicity. Next slide. So when using NGS, there is always a trade-off between using whole genome sequencing, whole exome sequencing, or targeted gene panels, or even single reads. And this trade-off will affect the cost, the time, the coverage, and the read depth. Of course, when we do whole genome sequencing, we are covering the whole genome, but at the same time, the sensitivity and the read depth will be very small. We can always achieve around 30x coverage, but that's not enough sometimes to detect mutations that are present at a very low allele frequency. However, if we use gene panels, the coverage is small. We are targeting a small number of genes. I mean, the portion of the genome that's covered is small because we're targeting a small number of genes, but the depth of coverage is very high, and we can achieve as much as 2000x. So mutations that are present at a very small allele frequency can be easily detected. Exome sequencing, same thing. You are covering more genes. You are covering 2 to 3% of the genome, but the sensitivity and depth of coverage can be small. It can reach about 50 or 70x, which cannot be enough to find allele frequency mutations that are represented at a very small allele frequency. So the first challenge with using NGS is to be able to determine, to make the trade-off between the time, the cost, the coverage, and the read depth. Next slide, please. So when comparing NGS tests and non-NGS tests, NGS has multiple advantages and superior qualities compared to other methods. NGS allows in one single run to test for all types of variants. For instance, you can detect single nucleotide variations, insertions, deletions, exon duplications, gene copy number, and translocations. And current non-NGS molecular diagnostic tests fail to detect known genomic alterations in patients who in actuality possess these mutations. And I think this statement is mainly highlighted by Alexander Drilon's paper in lung cancer patients, where he showed that actionable genomic alterations were identified in 65% of lung cancer patients deemed without targetable genomic alterations by earlier extensive non-NGS testing. And furthermore, a large percent of these patients' genomic alterations have a targeted agent accessible through a clinical trial. And then they further went down to show that a number of these patients that were matched to an ongoing clinical trial achieved good progression-free survival or overall survival. Next slide, please. So besides these clinical benefits of NGS, NGS has also some technical benefits. For instance, most NGS assays would require as little as 10 nanograms of DNA, and that 10 nanograms of DNA can allow you to detect small single nucleotide polymorphisms, deletions, insertions, translocations, etc., whereas non-NGS tests like FISH or IHC tests require several histologic sections of the FSP specimen block for a single run. And then the other benefit of next-gen sequencing is the swift turnaround time for comprehensive DNA sequencing to provide actionable data to clinical physicians, and that turnaround time will be even shorter as the technology is improving and the cost of sequencing is dropping. Next slide, please. I think the most comprehensive study that provided the foundation for a framework for the molecular classification of AML came from the Cancer Genome Atlas Network study that was published in 2015 in the New England Journal of Medicine. And in this study, the authors analyzed the genomes of 200 clinically annotated adult cases of de novo AML and used either whole genome sequencing, whole exome sequencing, along with RNA and microRNA sequencing and DNA methylation analysis. A total of 23 genes showed on the right were significantly mutated, and those include the FLT3 gene, which was mutated in over 50 samples that were analyzed, the NPM1, the DNMT3A also were mutated in over 50 samples. Other genes like IDH1, IDH2, and RUNX1 were mutated in about 20 samples, and another 237 genes were mutated in 2 or more samples. Nearly all samples that were analyzed in this study had at least one non-synonymous mutation in one category of genes that are almost certainly relevant for pathogenesis. Next slide, please. So the organization of the mutated genes into functionally related categories revealed many potentially important biologic relationships, which provided a comprehensive foundation for an understanding of the genetic rules of AML pathogenesis and led to discovery of many of the targeted therapies that Dr. Hale was talking about and that were approved within the last few years. And these categories would include cell signaling genes like FLT3 gene, KIT, JAK2, MPL, transcription genes such as RUNX1, epigenetic genes like DNMT3A, cell cycle genes such as P53 and NPM1, cohesion complex genes, and splicing genes. So next slide, please. So besides the single nucleotide mutations, this study also, in this study, discovered 74 in-frame fusions here in green, and then many of them have previously been described, including PML, RARA, RUNX1, RUNX1T1, BCR-ABL, and they also discovered multiple fusions involving MLL genes. An additional 42 gene fusions in orange were also discovered during this study. Next slide, please. So regarding the current guidelines for NGS analysis for the diagnosis of AML, there are two institutions that provide these guidelines. There is the European Leukemia Network, ELN, which recommends genetic testing for all patients with newly diagnosed AML, and they have three different requirements. So the first one is to use conventional cytogenetics. Second, use screening for a minimum of 6 mutations in the genes we talked about earlier, like nucleoplasmin and RUNX1, FLT3, P53, and ESXL1. Also, they recommend screening for gene rearrangements. So all these fusions that we talked about, PML, RA, RUNX1, RUNX2, and BCR-ABL. The National Comprehensive Cancer Network is the other institution that provides guidelines, and they recommend also conventional cytogenetics, testing for a minimum of 9 gene mutations, including NPM1, RUNX1, FLT3, and also testing for gene fusions identically to the ALN recommendations. So both institutions do acknowledge that the recommended mutational testing has to be interpreted as minimal for daily clinical practice in order to accurately assess genomic risk and use targeted therapy where appropriate. And also, according to these two institutions, multiplex gene panels and NGS analysis may be used to obtain further information regarding prognosis, treatment decisions, and eligibility for clinical trial participation. Next, please. So there are a number of gene panels that are currently available for targeted sequencing by NGS. Those include the SureSeq MyPanel from Oxford Gene Technology, the LookVantage panel from Quest Diagnostics, the AmpliSeq myeloid sequencing panel from Illumina, the human myeloid neoplasm from QIAGEN, but there are a lot of, all the clinical labs these days, they have their own lab-developed panels. Here at Medpace, we have myeloid panel that has 54 genes that is very similar to the commercially available tests. Next slide, please. So there are huge differences in cost between using whole genome, whole exome, and targeted sequencing. So we went over those trade-offs earlier, but there are caveats to take into consideration when deciding which method to use. And these caveats or challenges become more apparent when thinking about using whole genome sequencing. And many issues can arise when you are using whole genome sequencing, including, for instance, accuracy. So even if you achieve a 99.99% accuracy, we are dealing with 3 billion nucleotides, so you would have about 300,000 errors per patient. So also whole genome sequencing is time-consuming, very expensive. The interpretation of identified variants can be challenging. Data storage and access in the medical records can also be a problem. Education of providers, patients, and public can be an issue. There are also issues related to consent and reporting. And the other big problem when it comes to doing whole genome or whole exome sequencing is the incidental findings. So incidental findings are mutations that you are not looking for, but you would detect when you are doing NGS. And some of these mutations can be trivial or indeed beneficial, but some are problematic. And occasionally we'll discover lethal or untreatable late onset conditions. And then there is the issue whether to report those mutations or not to report them. Next, please. So I would go over some specific situations that are related to using NGS in hemato-oncology. So the first challenge I want to present is how to discriminate between leukemia-related mutations from polymorphisms or passenger mutations. So what we're looking for are leukemia-related mutations, but occasionally we will have polymorphisms or passenger mutations, and then the challenge is to discriminate between those two. So the current approach is one is optimization of cancer-specific databases, including reporting of rare physiological gene variants. So if these databases are complete, it will be easy to go to those databases and then find which mutations are indeed associated with leukemia and which are actually just polymorphisms or passenger mutations. Implementation of novel bioinformatic algorithms based on prediction of functional impact will be very useful, and also using quantitative and dynamic variant allele frequency monitoring, which is separately and together with other mutations, will be very helpful in this situation. Next slide, please. The other challenge is how to discriminate between leukemia-related somatic mutations from pathogenic germline alterations. So now we're looking at two different things in the same patient. We're looking at somatic mutations versus germline mutations and the challenge of differentiating between acquired somatic mutations from germline pathogenic variants. So there are a number of approaches that one can follow. So the first one is to detect mutations, germline mutations in control samples. For instance, use skin or fibroblasts or saliva in genes that are highly mutated in EML, for instance, like RNX and CABPA. So we can also work that issue through medical family, a very thorough medical family history, followed by molecular genetic tests if relatives, in relatives if necessary. Also, there are situations where there is a high and stable variant allele frequency, for instance, between 40 and 50% at follow-up despite clinical response to treatment. That also may be indicative of germline alteration rather than a somatic mutation. Next slide, please. So the other issue is how to discriminate through genetic alterations from mutations that are associated with the technique that was used for NGS, for instance, mutations introduced during PCR amplification, during sequencing, or post-sequencing artifacts. So many artifacts do indeed arise during NGS library prep, sequencing, and data analysis. And in order to resolve those issues, there is a need for error correction using molecular identifiers that individually these would label original input DNA molecules, so you know which ones actually were original in the template that was used for sequencing from the ones that were introduced during DNA amplification by PCR. Refinement of error correction computational methods in post-sequencing NGS data analysis also is another way. Also confirming using Sanger sequencing, but this is not going to be very helpful if your allele frequency is below 20%, because sensitivity of Sanger sequencing is not that great. Next. Another challenge with using NGS in immuno-oncology studies is limited sensitivity of NGS for minimal residual disease assessment. For instance, mutations detected at diagnosis may be re-identified at best to a variant allele frequency of 1 to 2%. So in this situation, again, error-corrected sequencing using molecular identifiers may be a good help. Complementation of NGS with other tools like real-time PCR and flow cytometry may be another way to do it. But again, MRD by flow cytometry or by qPCR has 10 times less sensitivity than NGS. So the best way would be actually to do a deeper sequencing and try to detect those alleles at very small or very low variant allele frequency. Next slide, please. So in conclusion, I want to talk about some, again, just to recapitulate on the advantages and pitfalls of NGS. NGS does allow a real insight into the biology of cancer, and NGS is important in predicting prognosis, which can help make treatment decisions. And the data from NGS can help build clinical and genomic databases that can be used for retrospective single institution or multiple institution studies. Next. So some of the pitfalls, always, it's a trade-off between time, cost, coverage, depth, etc. So Are you going to use whole genome sequencing, exome sequencing, targeted panels, or single gene? It all depends on the resources, the available resources, but also it depends on whether we have a good bioinformatics team, a good clinical and research team that will curate those variants, etc. And that would affect the quality of the results that were obtained. So we need expert annotation and analysis. So a good, like I said, a good team of bioinformatics and expertise with expertise in mutation annotation, a good team of clinicians and basic researchers around the table to decide about those variants, whether they are pathogenic, likely pathogenic in certain significance, or if they have no effect. Certain mutations are not assessed well in NGS, and that's from my talking from experience. For instance, like the internal tendon duplication in FLT3 and duplication in color gene, which are very important genes in the myeloid panel. Those duplications are usually hard to derive just from NGS data, and there is a need to develop to use a parallel method to detect those duplications, or develop new algorithms that would allow that detection to be done in a bioinformatics way. Finally, the NGS results are not the whole picture for prognostication of or prediction of in EML. Other factors such as age, the CBC counts, large indels, insertion deletions can be as important or even more important than the information provided by NGS. Next. So just to summarize all these things that we talked about, NGS application in targeted therapy, especially in AML. In the last 5 to 10 years, NGS has been introduced in most specialized hematologic laboratories with various NGS panels. NGS will pave the way towards a more detailed clinical evaluation and optimized therapeutic decisions as NGS costs are rapidly decreasing and technology is rapidly improving. However, changes remain before NGS becomes routine. We talked about cost and reimbursement, data analysis and storage, variant interpretation, limited sensitivity, for instance, in the case of MRD, and other technical aspects of testing that introduce artifacts that may interfere with the results of NGS. All these things can affect the results.
Currently, one of the biggest challenges in conducting clinical trials for treatment of patients with AML is a competitive landscape, which includes 8 new targeted agents approved by the FDA within the last 3 years. Additionally, there are currently 541 actively recruiting trials for the treatment of patients with AML. As such, both investigators and patients have many treatment options, and this is further limited by the rapidly increasing smaller group of patients defined by molecular abnormalities. In addition to the competitive landscape, there are protocol-specific challenges which make it difficult to recruit patients. Specifically, protocol eligibility criteria and complex or burdensome procedures need to be considered carefully, as these will influence interest from both investigators and patients. Furthermore, the availability of published data supporting the therapeutic target, safety, and efficacy will also influence interest from both investigators and patients. Time is also a consideration given the need for investigators to match the patient's AML or abnormality and driver mutations with a targeted therapeutic agent. Successfully executed clinical trials in patients with AML include a physician champion, in addition to enthusiastic investigators for a therapeutically targeted agent. As such, site investigator selection is a critical step in operationalizing a clinical trial for the treatment of patients with AML. Importantly, feasibility of potential sites and investigators should include an assessment of the number of patients, competing trials, and startup timelines, with the goal of selecting the right mix of sites to support the successful execution of the trial within the intended timeline. Additional consideration should also be given to sites which routinely characterize abnormalities for the intended therapeutic target. This assessment may occur locally at Foundation 1 or through a master protocol like those used by M2GEN and Beat AML. Further consideration should also be given to recruitment materials intended to educate the patient on the protocol and to provide travel reimbursement, which will ease the burden of clinical trial participation on the patient. Successfully executed clinical trials in patients with AML include simple protocols in which all complex and burdensome procedures are removed or limited, and if included, support the objectives. Furthermore, protocols with published data supporting the molecular targets, safety, and efficacy are more compelling to investigators and patients. Importantly, it is not a good operational strategy to execute a clinical trial for the treatment of patients with AML without published data. So in addition to operational logistics, successful execution of clinical trials for the treatment of patients with AML is predicated on both relationships and expertise. Specifically, there are 5 stakeholders which can influence the execution of a clinical trial, and any one of these stakeholders can be rate limiting. In contrast, and preferably in collaboration with these stakeholders, will support successful operational execution of the clinical trial. Therefore, it's important to ensure that experts in the treatment of patients with AML are included at the investigative sites, in addition to the sponsor and CRO, with an emphasis on the medical monitor, nurse practitioner, clinical trial manager, and clinical research associates. Furthermore, best practices should be considered, including the selection of enthusiastic investigators, a well-written protocol supported by published data, and the inclusion of procedures, and ideally those which support the objectives and are not complex or burdensome to the patients.
Well, thank you very much to all of our presenters for that very insightful presentation. At this time, I'd like to encourage the audience members to continue submitting their questions or comments right now using the questions window for the Q&A portion of this webinar. And to lead the Q&A, I'm going to bring back our speakers to facilitate that.
Okay, we've got several interesting questions here. The first one will be for Dr. Bahasi. One of our questions is, how do we make sure that the data generated by NGS is reliable?
That's a good question. So first of all, the current technology is really very good. And NGS workflows are very tightly quality controlled at every level, from DNA use to the library preps to the run quality on the instrument, to best practice data analysis pipelines, to strict guidelines for variant calling and interpretation that are usually based on the ICMG guidelines. Furthermore, most pathogenic, likely pathogenic invariants of certain significance are already well described in existing databases such as CleanVar. However, every variant still needs to be very carefully analyzed and interpreted.
Great, thank you very much for that response. A second question, one of our viewers asked, could you provide more information on CPX-351? I'll take that one. A CPX-351 is a liposomal combination of daunorubicin and cytarabine. Daunorubicin and cytarabine are the 2 agents that are traditionally given as induction chemotherapy, although they're given separately. For this medication, they are bound into a lipid micelle so that they're delivered in a constant molar ratio of 1 to 5 for daunorubicin to cytarabine. This allows a very controlled delivery of the chemotherapy drugs, and we know from preclinical animal models that the 1 to 5 molar ratio seems to have the best anti-tumor ability. The liposomal formulation of this agent is administered intravenously. The liposome is thought to enhance the stability by allowing it to escape metabolism and elimination in the body prior to be delivered to the malignant cell. And we also know from animal studies that the liposome is preferentially internalized by blasts as opposed to other hematopoietic cells. And this also allows it to bypass many of the resistance mechanisms that leukemia cells develop, such as the P-glycoprotein export system. And ultimately, this leads also to a longer half-life for the compound. So there's several advantages to this product. And then I think the last question that was submitted was can you provide more information on companion diagnostic testing? What I would just say about companion diagnostic testing is that the definition is that a companion diagnostic is a medical device, typically in vitro, that will give additional information for the use of some corresponding drug. The big point here is that it provides the physicians caring for a patient the ability to determine if a particular product will benefit a patient or if the patient is unlikely to benefit from it based on the cytogenetic or molecular entity tested. It's important to note that the FDA has issued several guidances on the development of companion diagnostic products. I think 3 over the last 5 years since 2014, and the ultimate one of the take-home points, they recommend early development and contemporaneous development of the assay along with the any investigational product, as the companion diagnostic often impacts the labeling. And now I'll hand it back to Candace.
Well, thank you very much for those answers. We've reached the end of the question or answer portion of this webinar. If we were unable to attend to your questions during the Q&A, the team at Medpace may try to follow up with you. If you have further questions, please direct them to the contact information showing on your screen. I'd like to thank everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Now please join us in thanking our speakers, Dr. Gregory Hale, Dr. Jeffrey Vassallo, and Dr. El Mustafa Bahasi, and feel free to share the recorded version of this webinar when it becomes available using the link in the chat box. We hope you found this conference informative. Have a great day, everyone.